All right, I know nobody's on here right now, so what I am gonna do is, do 5 minutes. So I'm gonna go let my dogs in and stuff, and come back here in 5 minutes. If you guys are watching this video, come back in 5 minutes. If I didn't delete this part already, so, maybe if somebody shows up or not, and then I'm gonna get started. I got a little bit of a late start this morning, so I just need 5 minutes, and I'm glad nobody's here, so I can take it. So I'll be right back. Hey, Valerie, sorry, nobody was on here, so I stepped away from it. Oh, let me go grab my iPad real quick. So, mm, okay. Well, let's get started. Let's do this thing first. Okay. So this study session is going to be covering chapter 10, which is the metabolism chapter. And so we're gonna get into that. So chapter 10. Okay, so to start off, and I was hoping that I could just pull it up on my iPad, but apparently it's like being weird. Oh, this way. I don't know what's, they bark at anything that isn't like normal sound. Something like the wind blew or something, and now they're barking. All right, so chapter. All right, there's within metabolism basically dealing with all of the chemical reactions in the cell, literally all of them combined. I don't know what I was thinking, typing with these nails on. So metabolism will be all of the chemical reactions in the cell combined, and we can even break that down further into catabolism and anabolism. And now we remember that catabolism, and like the way that I always remember it, cats being destructive, they're going to tear things apart. Catabolism deals with the breaking of bonds, and therefore turning larger molecules down into smaller versions of those molecules. And then anabolism is going to be making of bonds. What are modules, guys? So all of these chemical reactions are going on in the body. They all require energy, and some of them require help in order to occur. And it's one of the things that helps the most probably in our cells by far, of course, is going to be the enzyme. And enzymes are basically organic catalysts. So literally chemically organic. And a catalyst is anything that helps overcome the activation energy of a reaction. And while that might seem a little vague at first, it's basically just any energy that you just any energy that you need in order for the reaction to occur in the cell. So that's all that is. It's not meant to be a big mystery of, what is going on. So organic catalysts, which help overcome the activation energy of whatever the chemical reaction is. It helps it go faster. It helps it need less ATP in order to occur. And enzymes also, one of the best things about them is that they don't get used up. They can be reused pretty much indefinitely, as long as they're still maintaining their structural integrity from the chemistry around them. So we know how important those hydrogen ions are going to be in the environment. So yeah, organic catalysts, which help overcome activation energy, and they can be used over and over again. Used and aren't permanently altered. Okay, cool. So that's basically that. Enzymes, they act on a molecule. So if you are DNA polymerase or whatever, you have something that comes and binds in your active site that is called a substrate, that thing that binds there, and that's the thing that you're actually acting on whenever you are doing this stuff. So that substrate that's going to be the molecule the enzyme actually does the work on. I can type pretty fast normally, but with the nails, I haven't like adjusted typing with the tips of the nails. Okay. There. Okay, so substrate is a molecule the enzyme does the work on, and it binds at the active site. Finding the active site of the enzyme. Some enzymes need help in order to perform their work. That just means that there's other molecules that they get involved with. That would be make you, if some enzymes are just plain, they'll just do their work without any help, and that's great. That's just enzymes, and that's wonderful. Enzymes that do need help, however, they're going to have these things called cofactors that are going to help out with them. And if that's the case, then we know that there's got to be part of the enzyme that's just the enzyme, and part of the enzyme that's the cofactor. And they, of course, have to have their own names and all of this. So the holoenzyme, the way that I like to remember it is it's the whole enzyme, the whole functioning enzyme. This is going to be all of the parts together that are actually going to make the working enzyme. It's just me here today, so anybody will watch me. Come on. No, I know. Oh, there. Geez, so small, so loud. Okay, all right. Okay, so let's break it down. We have the apo enzyme. This isn't too many parts, so thankfully there's that. You have the apo enzyme, which is the actual like protein or enzyme portion of the holoenzyme. And then the other part is the cofactor. And this is just the part that is the added part. That helps the enzyme, the holoenzyme, do work. Sort of like when we think of iron and hemoglobin, right? Not cofactor B. So within cofactors, we have just things we would call cofactors, or things that we would call coenzymes. Ah, loud. Small. It's so stupid. I just see the like little black shapes move in the background. That TV behind me is massive. So like when I see them go, it just looks so silly. All right. Stupid. This is usually, the cofactors are usually, this is a big broad term for any cofactor. So this would be any, we'll say any added molecule. But then coenzyme is the more specific term. So this will be the organic or carbon-based molecules. So this would include things like usually our vitamins and stuff like that. So our cofactor would be things like metallic cofactors, like iron and stuff. I was going to say, because the vitamins, of course, if we're talking about vitamins and them being organic molecules, they still fall within the category of cofactor, right? But they just also fall within a more specific category of coenzyme. So, but what would we put the inorganic molecules that are cofactors into? Just cofactor. So that's the point there. Let's go back. Terms. There's a lot of terms in this one. Okay, so that's good. We're at that part. We're at now denaturation. So let's take that back a step. Enzymes. Enzymes depend quite heavily on the shape for their function. And this depends like largely, very, very much largely on the hydrogen ion environment around that that the enzyme is within, right? So so the shape depends on the hydrogen bonds in the protein structure. And these bonds are greatly affected. Wow, no. By the hydrogen ion concentration, so the H plus concentration with the little brackets like this. These are the concentration. You have long nails. Oh, all right. So clearly things like pH and stuff like that. You put it like this and structured. There we go. Okay. So for enzyme structure, enzymes depend on their structure and shape or function, and that depends on the hydrogen bonds and the protein structure, and they're greatly affected by the hydrogen ion concentration just everywhere around it. And so pH, temperature, usually high temp, hydrogen. I mean, there's a lot of other things that can affect the shape of the protein and the hydrogen bonds, but the ones we talk about the most are definitely pH and high heat. So I would really just focus on that one, but that can cause can disrupt those hydrogen bonds. So that change, the change in the chemical structure, that overall like tertiary, secondary, all those structures, that change in those structures causes the enzyme to not be able to work anymore because of the shape change. Now, its chemical makeup is still exactly the same. So that's the important thing to remember. We haven't changed it chemically at all by putting a lower pH or a higher pH or anything. There's been no chemical actual reaction going on other than disrupting those hydrogen bonds. So we haven't changed anything. We haven't removed any amino acids or changed them or whatever. But still, because the hydrogen ion concentration and the effects of the hydrogen ions, as well as breaking those hydrogen bonds with heat, those things disrupt that overall shape. And when we have disruption of the shape of the protein and that affects its function, then that is called denaturation. Denaturation, disruption of enzyme structure and function. Often due to effects on hydrogen bonds. That's okay. So let's talk about the reactions that our enzymes can support. We'll say enzyme-driven reactions. And we had those 6 enzymes classes or whatever that I told you guys that you needed to know, but you don't need to know all of them. So there are 6 of them, yes. The only ones that I care about. Let's go over the ones that we would potentially get tested on. The ones that I would ask questions about if it was going to be any of them. So that would be oxidoreductases, transferases, hydrolysis, sure. And ligases. So these are the 4 that I would be most likely to ask about. So let's go through what they are. In an oxidoreductase, where this involves moving electrons around. So electron exchange. And what we would call those reactions, they're redox reactions. Reduction and oxidation. We'll go through those in just a second. We have transferases that are going to be moving groups from one molecule to another. By groups, I mean chemical groups. Okay, next we have hydrolases and ligases. They kind of go together. But like hydrolases, I know it sounds like a strange word, we haven't heard it before, but we learned about these in the lab. We talked about hydrolysis or starch hydrolysis and gelatin hydrolysis and urea hydrolysis. That's what this is. Hydrolases are involved in hydrolysis and they'll hydrolyze things. So that's what's going on here. So what typically is going to be happening in those cases is breaking bonds. This usually involves adding water. So if we're talking about hydrolases, what they do, they hydrolyze, right? It's hydrolysis. So hydro is water and lysis is breaking apart. So we are usually breaking bonds by introducing water. So that's how that works. Adding water. Water is not the product. We're adding water and breaking water apart in order to break those bonds. It's almost like you're introducing water. You're trying to break two things apart, but you need to seal the ends in a way. You can't just like leave them chemically open. So the things that you're using is you're taking water, breaking it apart, and sort of using it like a patch to staunch the, I don't want to say the bleeding of the molecule, but to basically take up those open-ended bonds and whatever. So we're breaking water in order to break those bonds. So we can add water in there and use it as like a finishing reagent, basically. So that's the idea of how that works. Ligases work the opposite way. We are building bonds. Water is typically a byproduct. Okay, cool. So let's come back to energy and let's get back to that later, but let's talk about the redox reactions first. And we'll do this underneath that oxidoreductase area so that we can just apply that to nearly we understand, or at least have introduced ourselves to. So a redox reaction. Redox reactions are going to have oxidation. Let's do reduction. Reduction and oxidation. Always both. You never have just one. So, so for reduction, you will have loss of electrons. Or however you want to look at electron donation. This is how I remember it, which is, Leo. Gain of electrons. Yeah, now I'm writing it out. I should have freaking wrote it out before I started typing gain. Gain. See, even I have to write it out before I get started. I try to think through it, and then I like suddenly hits me. I'm like, oh wait, Leo says GER. So Leo says GER. So the oxidation is loss, and reduction is gain of electrons. Rewrite this and get it out there before I think too far ahead, because this stuff confuses me too. Sometimes not so much confusing. It's like once I have like Leo says "ver" written on my scratch paper, that I'm good to go. But I thought I could just think through it. I don't know why I fool myself into think through chemistry ever. So yeah, lots of electrons. Leo oxidized. So that is going to be electron. For this one, will be 2. Okay, that's better. So we have reduction is gain of electrons. Oxidation is loss of electrons. Loss of electrons, Leo, Le. O is oxidized. And then GER is gain electrons reduced. And that's how I remember it, or you can, of course, do like what the book, I think what the book suggests, which is oil rig. So this would be rig. Yeah, so rig is reduction is gain, and then oil is oxidation is loss. And then you would be able to help hopefully be able to use those little acronyms or whatever to remember, because I certainly depend on them, like obviously more than you might think, to remember the difference. I'll just be having like just talks about this like we are now, and I'll have to like remember by thinking Leo is Gur, and so that means in the middle of me talking about it. So that's one of those ones that I would definitely be writing on my scratch paper. But that's me. So anyways, that is really, I don't know why, we don't need to know anything else beyond that about oxidation and reduction, just where the electrons are moving. So if you are being oxidized, then you are losing your electrons. If you're being reduced, then you are gaining electrons, and they're always going to come in pairs. So it's redox pairs really more than it is anything. One atom is always going to be oxidized and one is going to be reduced in these reactions. You don't ever just have one. So there's that. But our best example was of chlorine and sodium in sodium chloride. Sodium becomes positive and chlorine becomes negative. We know that well enough. That's pretty well, established information. Come here, meatball. Meatball boy. That's pretty well established information. So the reason that sodium is positive is because it lost an electron. The reason that chlorine is negative is because it gained an electron. So if chlorine is negative, gained an electron, it was reduced. And it is that simple, and that really is basically how it will work in those types of reactions. Hi, spaghetti. They're so tiny. See them in the background. You guys coming in the background, little guys. So anyways, happy with that. Let's get into making empathy, shall we? I'm gonna start a new page for this one so that we can separate it out from all this like background of what an enzyme is and how it works and all that. So. Alrighty. So how do I want to start this? I guess we'll just go through aerobic respiration because it's just the next thing on the sheet. But first, of course, the most important thing to understand about all of this is the cell source of energy. ATP is that. So this is adenosine triphosphate. Is the source of energy. Cell. It's involved in any chemical reaction that needs energy in the cell, almost all of them. So that's basically the whole point. This is the big daddy. This is the point of going through glycolysis or going through Krebs or going through oxidative reduction or any of that. Whole point of those things is to make ATP. Okay. So that's aerobic respiration. I said, let me find this before else, like it just occurred to me. I want to find this chart. No. A lot more going on here. Here it is. This is the one. Okay. I want to add this one in. Just briefly, I mean, not briefly, but take a look at this really quick. Okay. This is so far down. What did the I just pasted it. Where did it paste to? Just the ether, I guess. Where did it go? I don't know where it pasted this. Okay, I see it here, but I also see like, where did my text go? Oh, it's under it. Hmm. Okay, let's move that. Okay. So with the image, this image is showing us basically a layout of the aerobic respiration versus anaerobic respiration versus fermentation, right? And so we can see quite clearly here. And I mean, honestly, this is all you need to know about aerobic respiration. I'm not really sure that we need to know a whole lot more other than a little bit more detail about maybe that electron transport chain. But what we do need to know is that in glycolysis, we are making a little bit of ATP and we are making a little bit of NADH. So that's our electron carrier. That's our energy product, which is the goal. You know, ATP is the goal of everything, of all of this, but it doesn't make very much. I mean, look over here, only 2 ATPs from glycolysis. And so when we look at that part of the reaction, glycolysis, we see that we have that present in anaerobic as well as fermentation. Those are different things. So anaerobic bacteria can never use oxygen. So they will only ever make their energy without oxygen. Whereas when we talk about our fermentators over here, these guys will use oxygen when oxygen and the other things that they need are available for aerobic respiration, but they'll be going through aerobic respiration. They won't be going through fermentation when oxygen's available. Because clearly, look at this. I mean, this is aerobic respiration over here. Anaerobic respiration over here can get up to 36, depending on the reaction we're looking at. But fermentation sucks. Like it just doesn't make very much energy as a net product. But that's because all they're doing is glycolysis over and over and over and over again. And all you get out of glycolysis is 2 ATP net. So that's why there's such a problem in fermentation as far as making energy. But to them, that's just a temporary thing that they do when they don't have oxygen. And so that's the only time they have to worry about that. They're only ever fermenting when there is no oxygen. If there is oxygen, the fermentation bacteria will just go through aerobic respiration. Aerobic respiration is also known as cellular respiration. It's the same thing. And the organisms, the fermenters that undergo fermentation and can switch to aerobic respiration, those guys are called facultative anaerobes, at least in the bacterial world. So we'll get to that, I think, in a little bit, but I like this chart because it kind of lays it all out. Nice big yet. Yeah, I know, really cute little baby. I know. I want more attention. I promise I'll play with you later. But yeah, so that's that. So in the aerobic and the anaerobic respiration pathways, we have Krebs cycle as the second step. The Krebs cycle is actually it's the same. It's the exact same in both of these guys. So there's no difference there. And then when we get into the electron transport system, the concept is the same. So they're going to be moving electrons down through cytochrome molecules, but for the anaerobes, they don't have oxygen as their final electron acceptor. They just have anything else. So it could be, well, go look at that. Nitrate is on there on that list. Why do you think we test for that in our lab test? So it could be nitrate, it could be sulfur compounds, it could be carbonate, it could be anything, but it's not going to be oxygen. So that's the important thing with that. So, are you cute? You are cute, aren't you? All right. So that's a good place to start. So that's our kind of general overview of what we're about to get into. Okay. Next. We can come back to this if we need to. A new page. Let's do that. What we are working on. back up so I'm staying on track. Okay, so let's do it. We're going to focus on aerobic respiration because that's what our cells do. And that's the primary like thing that everything else is going to be built and based off of. Even though a lot of bacteria undergo fermentation, the fermenters are going to go through aerobic respiration. And the concept, whether you're a bacteria or a eukaryote or whatever, is the same. So aerobic respiration first. It's aero because it involves oxygen. Okay, there's three major steps in aerobic respiration. There is glycolysis, Krebs. It sounds like my neighbor is mowing their yard. They don't do that. They don't mow their yards. So this is like a miracle. Ours is like, we mow every week. Theirs is like almost up to your knees. Like, so to hear the lawnmower suddenly is just like, is that real? Watch them knock like a rock through my window. I'm sitting right next to like that side yard. So, it was like a surprise. Anyways, good. I'm glad they're mowing their yard. I don't care. We don't have an HOA. I don't care. You do your business. I'll do mine. If it starts affecting the worth of my house when I'm trying to sell it, then that's one thing. But otherwise, I just don't care. Couldn't possibly be bothered to care. So, ETC, electron transport sheet. All right, so those are the three steps in aerobic respiration. Let's go through them because why wouldn't we? So this is easy enough. In glycolysis, this is just breaking down glucose, which is a 6-carbon sugar. Okay, we're going to break that down into 2 pyruvates. And those are 3-carbon. Okay, so that's glycolysis. In glycolysis, while we are doing that, we make 2 ATP and we make some NADH, which is an electron carrier. And I'll just remind us of that. It's a electron carrier, and it eventually will shuttle its electrons through the electron transport chain. So we make this periodically throughout this whole reaction. So that's glycolysis. We break down that 6-carbon glucose molecule to make 2 of the 3-carbon pyruvate molecules. What am I doing? I just want to be sure I'm not missing something. Okay, then there's an intermediate step that you don't need to worry about. So I'm just going to list it here under the glycolysis in parentheses, but just that we know why I'm telling you it makes pyruvate here, but what enters Krebs cycle is not pyruvate. So actually, I'm going to put that in Krebs because typically we list it under Krebs. So first in the Krebs cycle, we have to prepare. The first step is to turn pyruvate into acetyl CoA, coenzyme A, but create a dog out there. Another one would die. Anyways, yeah, so we're going to turn pyruvate into acetyl CoA. We'll also, in this process of doing this, make a little bit of NADH. We will also be making, sorry, now weed eater time currently. It's like all I can hear. It's so loud in here. I don't even know. It's weird how loud it is, but yeah, carbon dioxide. That's what I'm trying to say. CO2. So that happens first before you even enter into the Krebs cycle. You'll have to deal with that. Why doesn't this update like at the speed that? I even try to click out of it so that it will update, and it still takes it so long. I can't control that. So right, so now I can actually go into the Krebs cycle, and essentially we're going to be turning that acetyl-CoA into electrons and CO2. Okay, so by that, our products are going to, actually, let me clarify it so that we just look like at that and see what our products are. I'll do that as well. I bet they paid some money. If it looked. I am not judging, believe me. But they just seem like those types of people that would wait forever and then just spring on having somebody mow it like once a month or something. So whatever. There's a lot of people that live there. It's like a 3-bedroom house, and it's a grandma, and she's taking care of like her own children, grown children, as well as like her teenage grandkids. You know, bless her heart, she's doing everything she can. I'm pretty sure she's retired, so she's just doing what she can. It just makes for once a month mowing, which it is way harder than once a week mowing. I'm just trying to find where I am. Sorry on my notes. I'm distracted by the noises. Okay, right. So we have our electron carriers. Let's just say that electron carriers. But these are going to be NADH and FADH2. We'll also make CO2 in this one. We'll make a little bit of ATP. I'm pretty sure that's all I care about. So we'll say like the big point. I will put in little asterisks at the top of these. The purpose. So that we can remember that the big purpose of glycolysis is to make pyruvate. The big purpose of Krebs is to make electron carriers. Or to put electrons in the electron carriers, you know, make them functional. Okay, so we've got our electron carriers and all of this, and even though we are making ATP, that's not the point of these steps necessarily. It's just the goal. And remember, you're going to turn through Krebs twice because you get 2 pyruvate from 1 glucose. So you get 1 glucose molecule, you'll get 2 pyruvate. Each pyruvate will go through Krebs. So he'll turn through that twice. And then now we have all of that crap that we've made from those products from glycolysis and Krebs, and we're going to go through the electron transport chain or electron transport system, however you want to call it. This whole thing is driven by a process called chemiosmosis. And that is negatively charged electrons drive the shuttling of those positively charged hydrogen ions or protons, however you want to call them, the H pluses. So negatively charged electrons are moving across the electron transport chain. Those molecules that it's moving through, that's allowing it to do all of this, they're called cytochromes, like cytochrome C oxidase, like we learned in lab. And cytochromes will get excited like the pigments of chlorophyll in plants will from the photons. And that will allow for movement of those hydrogen ions, those positively charged protons outside of the membrane. And then that will create that gradient. And that has its own term, and that's called the proton motive force. And it's specific to this situation. Just because you necessarily have a buildup of proton somewhere doesn't always mean that you have a proton motive force. But that is what is happening here. It's going to help drive other things that are going to be going on later. What are you doing? What are you doing? You're small. Come here. So small. Yeah, you're still young and curious. Your older sister just doesn't care anymore, does she? She does. Cool. So that is chemismosis and the proton motive force. That's kind of the whole point of what's going to be happening in what drives the electron transport chain in making ATP, right? Because you have to build up that proton gradient. So generated proton gradient causes built up protons migrate back into the other side of the membrane. I know, because you're perfect. I'm just trying to get it to like update on here. It's just so weird that it doesn't want to. Okay, generate proton migrate back into the other side of the membrane. And then protons, those H pluses, will do the migration. It wants to try to restore the gradient, right? That's always what the goal is. They want to make it equal on both sides. That's just what chemistry drives it to do, chemistry and physics. So we'll migrate across the membrane. Across the membrane to the other side through the only real viable channel that they can move through, which is ATP synthase. And the presence of the positive charge of the protons is what's going to drive the production of ATP by ATP synthase. So we know about the importance of protein shape, enzyme shape, especially, and how that really did something get delivered. I love it when things get delivered. Will drive basically the positive charge of those protons, shape alteration of the hydrogen bonds within ATP synthase itself. So it's not really a chemical reaction that's occurring so much. It's like a shift in how they're reacting to the environment of having those positives go through there. And when that happens, it causes an overall shape change. And that is shape change is what drives phosphorylation of ADP into ATP, adding a phosphate group onto ADP to make it into ATP. This is called oxidative phosphorylation. So we'll add a phosphate. And make ATP. And that process is called oxidative phosphorylation. Why is it called oxidative phosphorylation? Because this whole thing involved electron exchange to make this happen. So it's just driven by electron exchange. Yeah. I'm pretty happy with that. That's pretty good. That's pretty good. Pretty good little summary we had there. Okay, so we've made that, we've done that. Now we have a buildup. Now we have built up electrons, protons, electrons and protons, and ATP, right? So, but we're trying to use ATP, that's the whole goal. So electrons and protons as byproducts, if not waste, right? So what do we do with that? Well, we bring in oxygen, literal O2, which we breathe in. And then that'll combine with leftover electrons and protons. Make water. And that is why oxygen is the final electron acceptor. Okay. Yeah, that's it, man. And that was aerobic respiration. And I feel like we really did a pretty good job consolidating that. I mean, considering what I had to learn. So this is way better. This is the important stuff. This is the stuff we would want to know. Basically, there's 3 steps to it: glycolysis, Krebs, electron transport chain. In glycolysis, all you're doing is glucose, splitting it basically in half, right? That's the point, to get pyruvate. Then pyruvate turns into acetyl-CoA, so that in the Krebs we can shuttle acetyl-CoA through to make to basically peel off all the electrons we could possibly want, and steal those off. We'll make carbon dioxide as a byproduct. Shuttle those electrons through the electron transport chain, and then that will create that proton gradient, the proton motive force, via chemiosmosis to drive the creation of that force. And then when those protons come back in through ATP synthase, it makes ATP. We have leftover electrons, we have leftover protons, and so we get rid of those by splitting an oxygen and combining it with some electrons and protons, and to make water as a product of that. And that's why we breathe in oxygen. And that's why oxygen is our final electron acceptor, because that's where your very last electron goes. Okay. Cool. Let me make sure I'm not messing with them. I don't think that I am. And so where is all of this happening in the electron transport chain and aerobic respiration and Krebs and all of this? In the electron transport chain, in eukaryotes, this is happening in the folds, or also known as the cristae of the mitochondrion. In bacteria and archaea, this is going to be happening on the inner membrane, inner or cytoplasmic, same thing. So remember that the inner membrane or the cytoplasmic membrane of bacteria and eukaryote, or sorry, excuse me, bacteria and archaea are important, not just for containing the crap that's in the cell, but also for being the site of electron transport chain and other things as well. A lot of stuff is going to be happening on that membrane because they don't have membrane bound organelles to do that stuff. So they have to have that happen on their inner membranes. So there's that. Okay, let's move on. Which were obviously, let me just bring this up really quick. Electron transport chain. This one is going to make 36 to 38 ATP. Whereas if we're looking at in Krebs, we only make like two. I think we make 2 ATP in Krebs, and we make 2 with each turn of Krebs anyways, and 2 in glycolysis. But the big daddy, of course, can be the electron transport chain. That's another one that I often ask about on the exams. It's like, which one makes the most ATP? And it's going to be the electron transport chain by far, like big time. So that's pretty good. And then we will say, okay, it talks about amphibolism. So let me come back up to this first page. I'm sorry, I'm moving up here. Say amphibolism. And amphibolism is just combining making and breaking of bonds reactions. To basically, I don't like how that is worded, but I don't want to word it. It's basically like saying, like, if I go through glycolysis and I made pyruvate as part of like what was made, right? That's the whole point of glycolysis. Go through glycolysis, we make pyruvate. And then we would turn it in acetyl-CoA and shuttle it through Krebs. Or we could use pyruvate to make amino acids, or use parts to make, nucleic acids and other things to help build things in the cell, or make energy, or do these things. And there's all these different pathways that you can go down from certain molecules. It is not as solid as like, okay, you have pyruvate. This is what pyruvate does. Well, it can do a whole bunch of other things too. It just depends on what the cell needs. So it will pull from whatever area it needs to, depending on, does it need more energy or does it need more protein, stuff like that. So that's what amphibolism is referring to, but it links like it says in the study sheet, the energy and the intermediate byproducts with one another for the overall metabolism of the cell. Make and break bonds reactions. And energy production. To tie in all reactions within the based on the cell needs. That's fine. Okay, and then we had fermentation, which we kind of already touched a little bit on. So I'm probably not going to go over that one too much. I feel like the big point of fermentation is just understanding about how it's just glycolysis over and over again, but we'll talk a little bit about it. So fermentation is only glycolysis. And it'll only make, I cannot, only 2 ATP. Per glucose molecule that's entering in via that route. So that's kind of boring, doesn't make very much energy, but if you don't have any oxygen, and that's what you'll do. So happens in the absence of O2. And this is only done by the facultative anaerobes. And they will go through aerobic respiration if O2 is present. Seems simple enough, very confusing for a lot of people for the lab questions that we're dealing with this when we're talking about fermentation. The concepts seem to make sense until we start putting them into practice there. I mean, it isn't that much of a surprise, I guess. That seems to be the case with the tests in general, but yeah, so this is what we're talking about, fermentation. Facultative anaerobes want to use oxygen and make lots of energy, but when oxygen isn't available, when we take that oxygen away from them, they will ferment. And they'll at least make some ATP so they don't die. All right. They still are going to need a final electron acceptor. Why though? Why do they still need the electron acceptor? Well, if we go back to glycolysis and we look at glycolysis, we see we make NADH as one of the products that we have. So that NADH that we were talking about, that will, it's carrying an electron. Normally, we in aerobic respiration will take that NADH and put it in our big pile of NADH and say, this is all going into the electron transport chain so that we can make ATP. And you might think to yourself, so yeah, so why aren't they doing that? Like, why aren't they putting that through where they can get more ATP from it? Because they don't have oxygen to put at the end to take care of the electron buildup there. They have to take care of that somehow. So they can't put it through that. So what they do is they just have to get rid of it in glycolysis if they don't have electron. That's the whole reason this is even happening. So fermentation, we still have to get that final electron acceptor because we can't shuttle it through ETC. We can't do the electron transport chain where oxygen would be the final one. So most of the time they will use pyruvate. Pyruvate. And then this will be turned into, we'll use pyruvate into either ethanol or lactic acid. It could be other acids as well. And then CO2 as a byproduct. These are the most common products of fermentation, and we learned about these in lab, and we're all familiar with them. You've eaten yogurt and it has that tangy taste, that's lactic acid. If you've had beer and it has fizz in it, that's the carbon dioxide and the ethanol making the alcohol there. There's all sorts of uses that we can put this through, whether it is our bacteria that we're talking about for our facultative anaerobes, or if we're talking about like yeast that are going through fermentation. It doesn't really matter, but they all deal with it pretty much the same way, making the same products and that sort of stuff. So that's the idea with that. And we're almost done, I promise. So that's the idea with fermentation. What else do I care about you guys knowing? It says fermentation and oxygen is not available. We are breaking down sugars and carbohydrates into carbon dioxide and alcohol and or acid. That's pretty much it. We learn in lab, and we talk about it in the lecture, that it's not necessarily always just going to be lactic acid that'll get made. It just depends on the bacterium, because some of them will make different kinds of acid. And there's tests that we can do to determine if you just do lactic acid, or if you do lactic acid maybe with one other acid, or if you do other acids altogether. And those last two, combo or other acids altogether, that's the Voges-Proskauer tests. So the methyl red is the mix, and the Voges-Proskauer is the one without, like, it's like basically other acids altogether. We get a little bit of lactic in there, but it's usually a whole bunch of mixture of randos. So that's what that's about. So that's what that's testing for. Cool. So let's just do photosynthesis really quick, and then we'll be done. see, photosynthesis. All right, we're saying light cycles use sunlight to capture energy to power carbon dioxide, fixation into glucose, which feeds respiration. Cool. So light dependent reactions and light independent. I call them the dark reactions. That's the old school. I just can't get it out of like the practice of saying it, but it's called light independent now, or Calvin cycle. Okay, so in the light dependent cycles, we have photons excite pigments. And then this is what drives the creation of that proton gradient. So we get a little bit of ATP here. So light dependent reactions, photons literally from light excite pigments that are in like the leaf or in the membrane of the bacteria. And they will then, when those pigments are excited, allow, instead of electrons moving through, is what my point is here. They'll excite the pigments with just photons, and that allows the proton gradient to be generated, and we can make a little bit of ATP without having to deal with. So this is, we've talked about aerobic respiration and like chemiosmosis and all of this, but this is relying on the energy of light. So we're not even dealing with having to introduce electrons or anything here. So it's pretty impressive, honestly. So that's the light dependent. The whole point of that one, of course, is going to be ATP. It doesn't make very much. You can't depend on the whole cell existing solely off of that for ATP, unfortunately. So the next step is the light independent reaction. And so what happens here is that ATP from the light reactions will be used to basically drive CO2, carbon dioxide, literally from the atmosphere, fixation into glucose. That's essentially the point of what is going on. Using the ATP to make glucose. And then oh my goodness. So the glucose made in this way will be transported through aerobic respiration to make as much ATP as possible. So way more ATP bountiful that way. I do just want to reiterate, when we think about plants, they do have chloroplasts for their photosynthesis. But they also have mitochondria for aerobic respiration. So they still undergo aerobic respiration. Photosynthesis occurs in chloroplasts. Okay, so that's fair enough. So in those light dependent reactions, so eukaryotes and then bacteria, In eukaryotes, we do this in chloroplasts. It's a membrane-bound organelle. It's quite a lot like a mitochondria. Has its own DNA, divides on its own, circular DNA like bacteria do. Has its own ribosomes and stuff like that. It's crazy, but that's part of the endosymbiotic theory. So chloroplasts, they are membrane-bound organelles. And then bacteria, what they do is they do this within the inner membrane or the cytoplasmic. Okay. If it does make oxygen as a byproduct during this whole process of making glucose and all of this, then that is called oxygenic, and that's where our atmospheric oxygen comes from. So, yeah, I mean, I don't know what else to say, but yeah, that would be using it during the light independent reaction. Yeah. Sorry. Oxygenic photosynthesis, where two comes from. Photos. Only in some photosynthesis. I mean, it's most photosynthesis. I don't know how to word it, but not all processes of photosynthesis create oxygen. There are types of photosynthesis that don't have oxygen as a product. They make weird stuff instead. So, but yeah, if it does make oxygen, actually produced here, then that's fine. It's made in that part of the reaction. So, but yeah, that is pretty much it for the rundown here. It always takes longer than I think it's gonna take. I always mean well. I'm always like, oh, we'll give a talk for like 15 minutes. And it never turns out that way. But anyways, I do have some slides pre-made with some open-ended questions, and I do have like those review things. So again, I'll just do the same thing that I did last time. We have this, once this is available for me to post it, it takes a while for it to process. But once this is available for it to process, then I'll post everything, the video, as well as the whiteboard link, as well as the PowerPoint for you guys to use for study tools as you're going through all of this stuff. So hopefully that helps some of you guys out there. Even if you're watching this after the fact, it's okay. So pretty much sums it up. Let me see if I can fix this. But yeah. So, any questions, Valerie?
No, thank you.
Awesome. Well, thanks for joining in, and I will, I guess I'll see you next week.
Thank you.
Bye-bye.